grade 12 learners. Welcome back to the Life Science lesson. My name is Samantha Ragunanan. This lesson is brought to you by the Gauteng Department of Education in collaboration with Saibono Discovery Center. So, learners, you may contact us on a number 079-454-9535. I'll repeat it, 079-454-9535. You may send WhatsApp messages, but no videos and no voice notes. Let's get started. So today our topic is protein synthesis. Myself and my colleague Agnes, we're going to take you through it, and after this you're going to be a master of protein synthesis. Now, like always, you need resources when you are studying for the grade 12 exam. It's not like the previous grades where you could get by on just studying or using one notebook or things like that. Here we need a variety of resources because we need to give ourselves the best possible advantage. And the good thing about grade 12 is that there are so many resources. There's the DBE study guides. There's past year papers, which is very, very important. When you are in grade 12 or any other grade, yes, you can study, but it means nothing if you're not practicing and learning the types of questions that they ask in the exams. You need the exam guideline, which is your go-to document. Now, the exam guideline will tell you what you need to study and how much of it you need to study. It is a very, very important document. Before you even go to your textbook, you should be going to the exam guideline. If you're not sure what that is, please consult with your educator and they will fill you in. And then there's the Learner Google Drive resource link. And I know that you are tech babies, so you know how to scan the code, the QR code, and access all the various documents. So it's not like you need to keep a whole big file of resources. You can just use your phone, and we know that you are visual learners and that you are able to look at the videos and you'll be able to learn much easily. And then, of course, there's the website, www.education.gov.za and www.pocketschool.co.za. Okay? Let's continue to the content weightings of the paper. Now, this is very, very important because the different topics have different weightings. What does it mean in English? It just means that on some topics, we ask less marks. On other topics, we ask more marks. So you need to know, because what happens if you're going to spend time and you're going to spend a whole 2 days or 3 days studying on a topic that only has 8 marks? Whereas you could have spent that time on a topic that has at least 50 or 40 marks in the paper. So when you start, you go to the exam guideline, you see which sections are in which paper and how much you need to know. Then you're going to go to the content weighting. So for the DNA, which you've touched on from last week, the weighting is 18%, which means that it is 27 marks. 27 marks is a lot. It could mean a difference between a pass and a fail, or distinction, or no distinction. So every single mark counts. Oh, we need to know the roadmap to get to where we want to be. Right. Now, last week you studied DNA and RNA, and you did DNA replication and DNA profiling. Now, the reason that I'm mentioning DNA is because it's a very, very important part of the process that I'm going to speak about today. The process that I'm going to speak about today is actually protein synthesis. So when it comes to protein synthesis, like the name intends, it's proteins, and synthesis means to make. It's to make proteins. And proteins, before we even want to know what is this process and what goes on, we need to know why. Why do we need proteins? Why is this process so important? Why are we studying it? Because when you understand why you are doing it, you will know how and where and all of the processes that are involved to get to the final product. So proteins actually help our body for the chemical reactions. It builds our immune system. Imagine if we didn't have a strong immune system. We'd be prone to all sorts of diseases. So that is why it is in our syllabus. It's very, very important. The process, now this is what we're going to cover. So this is remember I spoke about the exam guideline earlier on? This is like a cutout of the exam guideline. And this tells us what's in the exam guideline. So if you don't know what the exam guideline is, I'm going to tell you exactly what it entails. Protein synthesis. So this is what you need to know. A process. It's actually a process by which proteins are made in each cell of an organism to form enzymes, hormones, and new structures for cells. And we know that hormones are responsible in the metabolism, they're responsible in balancing of all the fluids in our body. So it's quite an important process, this. And then the process is divided into transcription, very, very important, where the DNA forms messenger RNA. So what's happening? Yes, you heard right. The DNA, prior to this, we learned DNA is this and RNA is this. But now what's happening is that the DNA forms messenger RNA. So then they become connected in some way. Remember we learned last week that the DNA is double-stranded, the RNA is single-stranded. Yeah, so now we're going to marry the two, and we're going to discuss the processes involved. Messenger RNA now has the coded message for protein synthesis. A coded message, that means something needs to be translated. If something is coded in a cryptic code like you see in the detective movies, they have to break the code, isn't it? And in order to break the code, for example, a different language, if I come and speak to you in a different language, I come and speak French to you, for example, and you don't know the language, you will need a translator. So do you see there's a code, and then the process of translation occurs, where the transfer RNA reads the code on the messenger RNA, and the amino acid links form a protein. So yes, it's a lot of words there, guys. But don't worry, we're going to unpack it and we're going to break it down so you understand it. You must study this because it is a common exam question. So while you are sitting there where you are, or standing, or whatever it is you're doing, I need you to have a pen and a paper, because you're not going to remember every single detail that I'm talking about. If I have to ask you grade 12s, what did you have last week Wednesday for lunch? You're going to have to think, oh, I can't remember. So the same, you are not superhuman, so help yourself. Take a pen and a page and start writing notes while the lesson is going on with your teacher as well. So let's look at the 3 types of RNA and their functions. Now, if you want to understand protein synthesis, if you know these 3, you will understand the whole process. Just knowing these 3. Do you see how important these 3 types of RNA are? Because each of them, I'll fill you in, each of them have a specific job to do. If we know what their job is, we will know the entire process and what their role is in. The thing with life sciences, that all of these topics are interrelated. Whatever we did for DNA, now we are using it in protein synthesis. Whatever we're doing in the section of DNA, we're going to use the same knowledge for genetics, for human evolution, all of that. So you cannot get by where you leave gaps in your knowledge. You have to know every single section verbatim. Okay, so let's talk about messenger RNA. The fact that it says mRNA, okay, M small m, that is an accepted abbreviation. They will accept it in the exam. So that means it's allowed. Okay, so messenger RNA, what do you think it means? Exactly what it says. It's an RNA which carries messages, maybe like a little gossiper RNA, okay? So it's an RNA which carries messengers. Now we know that messengers, now these days we got WhatsApp and we got Instagram and all of that. But back in the day, they used physical messengers to transport messages from one place to the other. So this messenger that we are talking about in the RNA, it is taking a message from one part of the cell to another part of the cell. So let us see what the accepted abbreviation is. Made up of many bases. We know that these are nitrogenous bases, isn't it? We know that these are nitrogen bases, and these are called codons. So that means they're going to code for something. It acts as a template for protein synthesis. It has the same sequence of bases as the DNA strand and has the gene sequence. They say it has the same sequence of bases as the DNA strand. Yes, we know that that can be true, but we also must remember at the back of our mind that RNA has uracil and DNA has thymine. So that is where they're going to differ slightly. So yes, they're going to have the same sequence, but not exact same nitrogen bases. Then we have transfer RNA. Now transfer small t RNA. So transfer with the small t is an accepted abbreviation as well. You can use it in the exam. So when you transfer something, so there I've got something and I'm transferring it to this spot. Where is it? It's moving from one place to the other. So transfer RNA, You see what I'm doing? I'm breaking up the term so that it makes more sense in my mind. Then I will understand the definition better. Bases are arranged in triplets called anticodons. So now notice, the one was codons, isn't it? And the other one is anticodons. So we know that these two are going to be connected in some way. It has a place for the attachment of the amino acid. Each transfer RNA codes for an amino acid. Anticodons are complementary to the codons. See, I wasn't lying. There's it. Anticodons are complementary. That means they're going to fit together and they're going to complement each other. Does it mean they're going to be the same? No, they're going to complement each other. RNA. So RNA, I think this is the easiest to, rRNA is the easiest. It's ribosomal RNA. So the R is for ribosome. It's an integral part of the ribosome. The organelle is responsible for protein synthesis. So what we know, what we took away from this is there's three types of RNA. There's messenger RNA, which is going to obviously carry a message somewhere. And we know that it has a code, it's called a codon. And we know it has triplet bases. And then we know the transfer RNA also has triplet bases, but it's called an anticodon. And we know that one is a messenger and the other one is transferring. So we know that something is going to move from something to somewhere. Okay? So we must have that image in our mind and the definition, and so we can move on to the next one. So this is the process of protein synthesis. Grade 12s, learn this diagram better than you know your name. If you remember nothing else from today's lesson, remember this diagram because it's going to come out in many, many different forms. They'll give you either this part, or they'll give you the second part, or they would ask questions based on it, or you'd need to label the diagram. So what I would suggest you do, make a study note where you redraw. When you are drawing and redrawing, for example, the DNA replication, it goes into your memory because not all of us can learn from notes. So if you're going to draw the diagrams, it's going to help you a lot. So there's transcription, okay? So when we talk about protein synthesis, we are talking about the making of proteins, and we spoke about the importance, okay? So let's talk about transcription. Transcription occurs in the nucleus. So this diagram here is the nucleus, isn't it? Excuse my handwriting. Right. That diagram there is the nucleus. We can see the nuclear envelope. We can see the nuclear pore. So that is where transcription takes place. Very, very important. If you are making notes, make sure you note that transcription takes place in the nucleus. They like to ask where it takes place. Whether it's transcription or translation, they like that question, okay? So you must know transcription takes place in the nucleus. To transcribe means to write, okay? So do you remember we spoke about in the old days they used messengers? So what they did is the kings or the queens or whatever would write a letter, and the person would take it on a horse or a donkey or whatever it is, and they would transport it to another part of the country or the land, and that's how the message was taken. So the same way, there's a message that's being copied here, and where this message is being copied from is from the DNA. So let's look at what happens. The double helix unwinds. Now we're talking about the DNA. There's the double helix. They're talking about this process here. There you have the DNA like this, the double helix. It unwinds, okay? Weak hydrogen bonds between the nitrogenous bases break. Does sound familiar? Yes, it's the exact same starting to DNA replication. But don't be fooled. It's a different process. Many grade 12 learners make the big mistake of confusing the two processes. So when I talk now, you should be writing. And the two DNA strands unzip and separate. Okay, so up until there, it's the same as DNA replication. No problem. We've learned this, we know it. This is where it changes. One DNA strand acts as a template for the construction of messenger RNA in the nucleus. So there's the nucleus. And we got one DNA strand acts as a template. Now, prior to this, what had happened is the double helix unwinds, and then both strands act as a template. But now we only have one which acts as a template. And then free RNA, not DNA, free RNA nucleotides from the nucleoplasm. We know that it's we call it nucleoplasm because it is the plasm or the liquid that is inside the nucleus. Hence we say nucleoplasm. This terminology and these steps, you must know it off by heart. Okay? You can't leave out anything. To form mRNA. So let's look, now that we spoke about the notes, let's look at the diagram, okay, to help us understand it. The double helix unwinds. There's the double helix, okay, and we can see that it is unwinding. So as the double helix is unwinding and the weak hydrogen bonds break, you know what happens? That messenger RNA sneaks in. Before the DNA can close again and replicate, it sneaks in and it starts to copy the code. So what happens there? The free nucleotides from the nucleoplasm, the free nucleotides from the nucleoplasm, they line up and then they start forming a template. They start forming from the template, they form the messenger RNA. And what happens is that the messenger RNA starts to look like that. Okay? So that's the messenger RNA. So what happens? We have DNA replication, the first two steps. The double helix unwinds, the weak hydrogen bonds break, and then one DNA strand serves as a template. So it will either be this strand or it will be the bottom strand. But in this case, we can see that it is forming from the bottom strand, okay? And it forms a messenger RNA, and the messenger RNA complements the DNA in the nucleus. So the message was formed. There we have, and now what do we call it? We call it a codon. So messenger RNA is called a codon. Why? Because it has copied the code from the DNA. So it's got the message, encrypted message. It's so exciting. So this is where the drama begins. So messenger RNA, after the DNA has already, before it replicates it copies the message. What does it do? It doesn't stay there in the nucleus. It goes out. And where does it go to? This section is actually the ribosome. Sometimes they don't, grade 12, sometimes they don't write labels and they don't say it's a ribosome. But you must remember, it moves from the nucleus to the ribosome, okay? So you can say, like, it's almost like when your teacher sends you to another teacher's class. Think of it, your teacher as Mrs. Nucleus. So Mrs. Nucleus sends you with a note, Samantha, go to Mr. Ribosome's class and give him this message. Okay? It's the same thing. So remember, Mrs. Nucleus sends a message to Mr. Ribosome. Okay? Mrs. Nucleus, Mr. Ribosome. Remember that. Message is going now. So that's what happens. So there we have mRNA. Okay? Remember we spoke about mRNA carries a message. Now what other RNA we spoke about? We spoke about this one here. Transfer RNA. As you can see, now what you need to take note of here is the diagram of the transfer RNA. It looks like a 3-leaf clover. It looks like a little curly diagram, but what we must take note of, and many textbooks do not have the exact same diagram, but what you need to know is that on the top, you will find the amino acid. On the bottom, you will find the nucleotide bases, the triplet pair. Okay, so what is happening? The transfer RNA is found in the cytoplasm, and what it does is you can see it's moving towards a messenger RNA. Something is going to happen, but before it happens, I need you to know the structure of the transfer RNA. It looks like a 3-leaf clover, and it looks like a plug point at the bottom, and at the bottom you got your triplet pair, not 1, not 2, 3 nitrogen bases that are there, that they carry to the ribosome. Now this is known as the anticodon. So this one here, this is known as the codon, and that is the anticodon. So the anticodon is going to, what happens here is that the transfer RNA actually goes and it looks for the appropriate and specific RNA nucleotide bases to pair with the messenger RNA. So let's see. Translation. It occurs in the ribosomes. The coded mRNA leaves the nucleus. There's it leaving the nucleus through the nucleopore to the ribosome. There's the ribosome, Mr. Ribosome. The transfer RNA picks up the amino acids and complement that complement the messenger RNA in the ribosome. So there we have the anticodon with the triplet bases. So as you can see here, this is UGU. So remember, in DNA, in RNA, we know that adenine pairs with uracil and guanine pairs with cytosine. Okay, so here, if you got U, the pairing would be A. If you got G, the pairing would be C. And if you got U, the pairing will be A again. So it's ACA. So where is this anticodon going to land? It's going to land here because there's it, ACA. Hence they put together. That is what happens in transcription and translation. So what we need to remember, we need to know where the process takes place. It takes place in the nucleus. Mrs. Nucleus sends a message to Mr. Ribosome. Okay? The messenger RNA copies, before the replication process can finish, it copies the message and it transfers it to the ribosome. What do we need to know about transfer RNA? Transfer RNA is basically like a taxi. When you get into a taxi, the taxi is going to a specific area. If you get into the wrong taxi, they just drop you there. You must choose the right taxi. Same way, transfer RNA taxi. It transfers, transports, transfer RNA. So the transfer RNA transports the amino acids to the specific codons on the messenger RNA. And that is the process. Simple, simple, simple. Sometimes we get grade 12s, when we see this picture, we say, oh, this is, we can't. This is too complicated. No, you can, because we've just broken it down. What do we need to know? We need to know the 3 different RNAs. So let's move to the differences. So both strands act as templates in DNA replication. And in transcription, the differences between transcription and translation, one strand acts as a template. They can ask you this. If you pay attention later on, there's exam questions that are based. They're going to ask you, what is the difference between DNA replication and transcription? And this is where you can score your mark. So if you learn this off by heart, great for you. Okay, so you must learn this. 3 nucleotides, it's so easy. 3 nucleotides, it's the same thing, it's just the opposite on the other side. 3 nucleotides from the nucleoplasm attached to each strand. 3 RNA nucleotides from the nucleoplasm attached to the template strand. See how easy it is. Whatever is on the left hand side, it's the opposite on the right hand side. But make sure you are comparing the same structures in the same line. You can't mix them up. Okay? Complementary base pairing occurs. A is paired with T and G with C. And then obviously the only difference in transcription is that A will be paired with U, because remember, in transcription, messenger RNA is involved. The 2 identical molecules are formed, and in transcription, a messenger RNA is formed. So what are we comparing? We're comparing what is happening in the nucleus, because transcription takes place in the nucleus. Okay? Let's move on. So now we're going to do a question. And I know you're going to do well. So what we do when we are approaching a question like this, especially questions that's got diagrams about processes, we must first look at the diagram. Don't look at the questions, okay? Sometimes it can just make you overwhelmed. Let's make sense of the diagram, and then we will move to the questions. So let's see. There we have, obviously we got something happening here. There's some pores. So this looks like the nucleus. So we're going to say that that is the nucleus. Remember Mrs. Nucleus? Don't go write Mrs. Nucleus. It's only nucleus in the exam, okay? There's Mrs. Nucleus and Mr. Ribosome. There's the ribosome there. And then the process. Now sometimes in the question, when we discuss the process, we can become confused, because the whole diagram, the label for the whole diagram, this whole thing is called protein synthesis. That whole diagram is called protein synthesis. The 2 stages in protein synthesis is process A, which we know is to be transcription. Remember transcription, to write message, you can relate the two. And the one in the ribosome, we know it is translation. Because remember, when something is written down, we have to make sense of it. We got the code. So where is the codon? There's the codon. What is the codon? It's the M. I can put a small m, capital RNA, because that is the accepted abbreviation. So there's the messenger RNA, and this diagram here. Remember, I said to you, all the diagrams are not the same. But the fact that you can see the triplet base pair here and something on the top, there's it, something on the top. We already know that these are amino acids. And we know that amino acids are bonded together by peptide bonds. Peptide protein, you'll remember it like that, okay? So I think we made sort of sense out of the diagram. So let's look at the notes. But be careful when they ask you for the process, look at what they are asking you, because there's many processes that's taking place here, grade 12s. The diagram below shows two processes that take place in all cells in the body. Provide a heading for the entire process illustrated. Notice, did they say a process? They said the entire. You must read very, very carefully, because sometimes they can trick us, okay? And that would be protein synthesis. Name processes A and B. We've done that already. We've already labeled the diagram. And obviously, the process A is transcription and the process B is translation. Now remember, they don't have to give you the whole diagram. They can give you only transcription diagram, or they can give you only translation diagram. Briefly describe the role of molecule C. Where's C? Let's look for C. We already identified it. We said it is transfer RNA, isn't it? Sorry, tRNA. And when they ask you, describe the role of a certain molecule, even though they're saying describe, the first thing you must do is you must name what organelle it is. And it is the transfer RNA. It carries a specific required amino acid to the ribosome. When the anticodon of the transfer RNA matches the codon of the mRNA. So that is basically what we've explained. And it's just a matter of us putting our answer into the questions. Let's look at the next one. The table shows different transfer RNA anticodons and their corresponding amino acids. Table 1, list of amino acids with the corresponding anticodons. You see this diagram? They like to ask that diagram. They'll give you the transfer RNA with the anticodon, and they'll give you the amino acid. And it's very, very simple, because they're giving you the key and you must just name the amino acid that is formed. Provide a label for the chemical bond D. There's the chemical bond D. Remember, peptide protein. So it is the what bond? It is the peptide bond, correct. Identify the amino acids at E and F respectively. So at E, let's find E. So the amino acid at E is G. Whoops. At E, sorry about that. E is GGC. Right. There we go. So if we're looking at here, it's GGC. So what we have to do is we have to take that, if we have to look at E and look at the triplet base pair and match it to what is there. And if we can look at this, we will see that it is proline. Can you see that? And then here you have UGC at F. And obviously UGC, let's find UGC there. You see how easy life sciences is? It's just matching columns, okay? So all you have to do is you look for that transfer RNA anticodon and you match it, okay? Now, another thing that I want you to remember, sometimes we get confused. Which one is the codon? Which one is the anticodon again? We become confused. What's a nice way to remember it is that A, okay, anticodon is on the top and codon is at the bottom. So A comes before C in the alphabet. So obviously the anticodon is going to be on the top and the codon is going to be bottom because A, B, C. Okay. So if you get confused like that, it's a very easy way to remember it. Provide the DNA triplet code that would have to code for codon G. So there's codon G. What does codon G here say? It says GCC. So what must we do? We must provide the DNA triplet code. So GCC the code will be for G. Remember, G pairs with C and A pairs with T in the DNA. So obviously GCC will be CGG because I'm going to rewrite it so you can see clearly. GCC. So what will G pair with? C. What will C complement? G. And C complements G. So there we have it, GCC. So we took that from there and we worked out the code there. Cytosine, guanine, and guanine. Okay? During DNA replication, one nucleotide might be placed in the wrong position on the forming of the complementary strand. This is a type of mutation. Name this type of mutation. What mutation is that? It's DNA. DNA is dealing with genes. It's gene mutation. Now, you might be familiar with gene mutation when you watch movies like X-Men and Spider-Man. Remember, there was a chemical reaction and that changes the structure of the genes. Okay? So that is why when you look at X-Men, they have the ability to stretch and they have got laser vision, is because the DNA sequence has been altered. So when you watch those movies again, you'll be able to understand it a little better now that you're a master in life sciences. Protein synthesis. Now this question was in last year's paper in the June exam. That's why it's so important to go through questions so that you know what they're going to ask you. Sometimes you can even predict the question if you get enough practice. So during DNA replication, if the DNA triplet code that resulted in codon G, so we look for codon G, there's codon G, okay? Were to undergo a type of mutation mentioned in the previous question, and the DNA triplet changes to CGT. So the triplet, so from GCC, it changes to CGT. What's going to happen? Let us explain the effect. So it was first GCC. Where did we get that? We got that from the question. Do you see that? On the mRNA, because this is the mRNA, okay? And it changed to GCA. So obviously if the codon is going to change, then it changes to GCA. What's going to happen? The anticodon is going to change. And then it's going to be CGG, okay? And that change on the transfer RNA changed to CGU. Oh, all these letters. Both anticodons, GCA and CGU, code for alanine and the same amino acid, which was in the table that we got before. So what would happen? The protein would not change. Now, this is another case of looking at the question and then trying to answer. I don't need to tell you what's going on here because by now you already know. So grade 12s, if we look at this process, the fact that I see already a T there, I know that this is DNA. I know that it's DNA replication. So do you see how they can give you a loaded question where they won't just give you transcription and translation. They'll throw in DNA replication to confuse you. And then it becomes, now this arrow is very important because something happens and we know that this is going to be messenger RNA. Because remember, messenger RNA sneaks in. But let's see. Identify the process at Z. What is the process at Z? It is obviously going to be transcription because messenger RNA transcribes the message, it codes, sneaks in and gets the code of the DNA. Molecule R. What is molecule R? We know if it's transcription, it has to be messenger RNA. Organelle Q. What is organelle Q? Because there's two organelles we're dealing with, Mrs. ribosome and Mrs. nucleus and Mr. ribosome. So if that is the nucleus, then this has to be the ribosome. Okay? Sorry about that. It's actually ribosome. And there you got the mark. Give the collective name for nitrogenous bases at O. We know, remember we said A comes, so this one is going to be the codon. So A comes on top, so it will be the anticodon. Are you with me? I hope you are following, because this is all work we've done in that main diagram. Determine the sequence of the nitrogenous bases at the area S. Where's the area S? Now look at this area S. So they want you to tell them what is the complement of T, what is the complement of C, what is the complement of A. Okay? We must be very careful, because the first thing we need to see, is it DNA or is it RNA? The fact that we see T here and the winding of the helix and all of that, it gives us clues. So we know that A is going to complement T, adenine, guanine is going to complement cytosine, and thymine, because it is DNA. So we're going to end up with A, G, and T. Am I right? Do you see how we can get a loaded question like that? Now, if you look at the second part, which strand 1 or 2 was used as a template for the formation of molecule R? There's molecule R. Okay? So they want to know which one, was it the strand or was it the strand that was used to form that? Now, we need to see what it would have complemented with. It can't be strand 2, because A and A won't complement each other. We know the complementary pairs. We know A and T, and we know G and C. So it has to be the strand, because if you look at it, A complements T, A complements U in mRNA, G complements C, and let's see, correct. So it would be strand 1. Which amino acid, 3, 4, or 5, was brought into area P? Where's area P? Let's locate it. It's almost like we're doing geography, eh? Where's area P? There we go. There's area P. Which amino acid was brought into P? So how I would suggest you do this, you do it as a process of elimination. If you look at the codon here, you got A, U, C. So what is going to pair with A? U, isn't it? What is going to pair with U? It's going to be A, and G is going to pair. So that one is going to go there. Okay? What is going to pair with this one? It's going to be C, C, A. So that one is going to pair with the second one. So obviously number 4 would be the one that is brought to the area P, and that would be correct. So what do we did there? What did we do there? We did a process of elimination by working out the rest of them, the code of the anticodon and the codon. Name the type of sugar that forms a structure at molecule R. Now remember, we have to be careful, because in RNA it's ribose sugar. In DNA it's deoxyribose sugar. And in this case, because we know that this is messenger RNA, it would be ribose sugar. You see how they try and trick us? But we are so sharp, we know the difference in the sugars. The diagram below shows the transcription through protein synthesis. So don't look at, remember, questions can become overwhelming. Let's look at the diagram and let's look for clues. Okay? The fact, let's look at this diagram. Now, obviously, we were luckier. They labeled it. There's DNA and there's messenger RNA. So we know that here we're going to have our phosphates, we're going to have our deoxyribose sugar, and we're going to have our nucleotide, nitrogen base. And there we have the nucleotide base, sorry. And then in messenger RNA, we know that it's going to be a single strand, and we know that adenine is going to pair with uracil. Okay, so let's see. Name the part of the cell where this process occurs. They're talking, what process are they talking about? We have to go to the question. It represents transcription. Where does transcription take place? The nucleus. Identify the sugar at X. We know that it is DNA, so it would have to be deoxyribose sugar. Don't be fooled. Okay, and then the nitrogenous base Y would be at uracil. There we've already labeled it. If we move on, tabulate the two differences between DNA and RNA. I'm not going to go into detail with this because we've already done this. But we know, I want to show you the mark allocation. We've done the differences, but remember, you have a mark for table as well. And this point must correspond with that point. So the strand, nucleotides, nucleotides. You're comparing apples with apples. Do you see that? Okay, so that is very, very important. And then we move on to the last question. I will read it. A gene, VKORC1, codes for the blood clotting factor in humans. The gene is made up of 163 amino acids. a mutation occurred that affected the amino acid 128 to 139. X-Men, remember Spider-Man? This is what's going on. The sequence CTG changed to CAG and TAT becomes TCT. This mutation has been transmitted as an autosomal dominant characteristic through generations. The mutation has resulted in resistance to warfarin drugs in humans. Warfarin is used in the treatment of thrombosis. Thrombosis results in the formation of a blood clot in the artery, and we know that this can be dangerous because it can cause a stroke and it can stop oxygen going to cells in the body. Warfarin causes the thinning of blood to break down the clot. Give one piece of evidence from the paragraph that shows that this mutation occurs in a DNA molecule. Now, why we've given you this today is to show you that there's many different ways they can ask you the question. So this is one of the ways where they give you an article. And we know that this is a DNA molecule because remember, the minute we think DNA, we must know thymine. So I'm already, even if I didn't read it or remember reading it, I'm looking at DNA, I'm saying it must be thymine. And then I go through the passage, I read the passage, and I look for the word, and there's it. Thymine is there, okay? Describe what is meant by autosomal dominant allele. So an autosomal, remember autosomes is the first set of chromosomes. It's a form of a gene and it is chromosome 1 to 22, the non-sex ones, okay? And it's always expressed as a phenotype and the individual in a heterozygous condition. So what we will do now, we will stop there. No, we've got one more slide. How the mutation on BKORC1 resulted in the resistance of warfarin in drugs. So there we have the mutation where the codon from GAC changed to GUC. Now if the codon is going to change, what does it stands to reason? If the codon is going to change, then the anticodon is also going to be changed. And in turn, what's going to happen? The amino acid is going to be different. So that is exactly what they are saying there. This changed the sequence of the amino acids and a different protein was formed. The effect of this mutation on humans with thrombosis. Now remember, they're not really too worried about thrombosis. They want to know that you understand what happens during a gene mutation. And in this condition of thrombosis, it is harmful because the blood clot is not broken down. It will lead to blockage in arteries and oxygen. That is how people sometimes they do not have proper brain function, or one part of the brain body goes lame. It is a very, very serious matter. So this section is a very, very important section. But we're going to take a break for now, and then my colleague Agnes is going to continue with the whole process of mutation and what it involved and how it affects the protein synthesis. So thank you for listening, and I hope that this was a fruitful session. Please stay tuned for the next part.
Good afternoon, grade 12 learners. My name is Agnes Moamelo, and I'm here to do the second session for today's lesson, protein synthesis. And thank you to my co-presenter, Samantha, who did very well in explaining what happens during protein synthesis. Right, on a lighter note, yes, grade 12 learners, we are on our 4th week of our academic year. And it's already hectic. And this means you need to set target for yourself to say, for this term, this is what I want to achieve. For the second, third term, this is what I want to achieve. So what is important again, you set goals, that target for the end of the year to say, I'm going to get 100%, which is very, very possible. And taking you through DNA code of life, yes, we are talking about the process of protein synthesis, which entails transcription and translation. Yes, like Samantha mentioned, you need to know those processes by heart according to exam guideline. So exam guideline should be your Bible for the whole year, because it is the one that is going to guide you on what to study, on what to prepare for the end of the year exams. Okay, right, for now we're going to look at the effect of mutation on protein structure, or we're looking at the effect of mutation on the DNA sequence. Right, if we look at mutation, you can ask yourself, what is mutation? What causes mutation? Even exposure to the viral or to the viral infections can cause mutation, right? Now, looking at the contents of the DNA sequence, and remember, even with those viral infections, they can lead, or they can end up causing what? Mutation that will lead to diseases like you talk about the cancer, you can talk about the asthma, and be careful for life sciences. We know we are next to cleanliness, isn't it? When you cough, you must close your mouth, right? When you sneeze, you must cover up your nose, because those droplets might infect the other person, and that can lead to what? Mutation within. That can cause some of the diseases, right? Effect of mutation on protein synthesis. Now you can ask yourself, what is mutation? And here we are saying gene mutation is the change in the DNA sequence. Change in the DNA sequence. Remember when we started in our last session, we mentioned that the DNA is double helix. The DNA is made up of what? Those nucleotides. Okay? So what you're saying here is changes in the DNA sequence is referred to what? To gene mutation. Right? So when those mistakes, we can call that mistake, we can call that a change, we can call that mutation. When it occurs, what is going to happen? So a gene mutation affects the arrangement of single or a few nitrogenous bases. I'm just going to go back to remind you about what we're talking about when we talk about nitrogenous bases. Think of adenine pairing with thymine, cytosine pairing with guanine. And do not forget, they are bonded together by what? Hydrogen bond, and not nitrogenous bond. So when mutation happens, it changes the sequence of what? Of few nitrogen bases. Okay? Now, what is going to happen? Just a quick recap again to say, because we're talking about changes in the DNA sequence, we're talking about the nitrogen bases. Also, because we're talking about protein synthesis, we're looking at how proteins are made. For a quick recap, we know that a group of those amino acids bonded together by peptide bond, they form what? a protein, right? So when we've got the same amino acid that may be coded for, and also they can cause no change in the amino acids or no change in the sequence in the protein, that will lead to the formation of what? The same protein. Contrary to that, when we've got different amino acids that may be coded for, which causes what? Change in the amino acid sequence in the protein, that will lead to the formation of what? A different protein. Okay? Please note that when mutation takes place, a mutation will not always lead to the formation of a different protein. Hence, we're saying if we've got amino acids, same amino acids that may be coded for, right? Then that will lead to that will cause no change to the sequence of the protein. Then we're going to have what? The same protein be formed. But what will happen if we've got different amino acids that may be coded for? What are we saying? They can cause change in the amino acid sequence in the protein, or they can cause a change in the amino acid sequence in the protein that will lead to the formation of a different protein. Hence, I'm saying when mutation takes place, it doesn't mean it will always lead to the formation of what? A different protein, right? I'm going to give you two scenarios that are going to help you to attempt questions on protein synthesis. Definitely you're going to get those questions in our paper too, right? So these scenarios will help you and this does not mean that the questions that I'm going to assist you with are going to be the same. The very same questions that you're going to get at the end of the year or in June exams. You might get totally different questions, but what I'm trying to say now, please master the concept, master your content, so that you are ready to apply to any question that can come across or that you can get in your exams, right? So first scenario, girls and boys, fortunately, examiners will guide you, will give you even tables where you are going to be guided. You're not going to start afresh thinking of scenarios. You will be given. So we've got a table that shows some mRNA codons and the corresponding amino acids. So these are mRNA codons. We've got amino acid. You even don't have to study the names of this amino acid because they will be given. Okay, so with the mRNA codons, they are given, and we even given the names of those amino acids, right? So a section of mRNA has the following base sequence and is read from left to right. Okay, so these are triplets. They are triplets. We call them triplets. We call them the codons. Now, if you look at those codons given, the first codon, we've got GAU, we've got CUC. You can read all of them. And we can refer to this as the original base sequence. Now, what you need to do if you are given this type of scenarios, you get your original base sequence, and then you compare it to the base sequence that is formed after mutation has taken place. Let me go back to it. This is the original base sequence. Now, after mutation has taken place, right, we're going to have a new base sequence being formed. It cannot be new for that matter, but you have to observe all those codons, if there are changes or not. Now, if you look at the original one, we've got these codons. A mutation occurred which resulted in the following base sequence on the mRNA molecule. You are told that after mutation, this is what you're going to get. Then, For you to score those marks, remember when I started, you need to set your targets, right? So you deserve every mark. Okay? Then you start to compare the original and the latest that was formed. You start with the first codon. Are they the same? Look at this one. GAU, GAU. They are the same. The original and when mutation has taken place. Compare the second codon. CUC, we've got CUC. They are the same. You are still safe. And then the third, GAC, GAC. They are still the same, isn't it? And then the fourth, we've got AGC, and then we've got AGU. This must tell you something. Okay, so it means on the fourth codon, the C was replaced by U. Then you start to ask yourself, oh, they have changed. Remember, I've mentioned that it doesn't mean after mutation, all the time you're going to get a different protein, right, being formed. Come to the fifth codon, AUG, we've got AUG after mutation. Original, we've got ACC after mutation, ACC. They are the same except on the fourth codon. So this means mutation has occurred. Why? Because C was replaced by U. Now, these are the steps that you need to, you should know all the steps when you answer those questions. Firstly, you compare the original mRNA to the one that has undergone mutation. We spoke about that one. And then identify the affected codon or nucleotide. We have said the nucleotide that was affected, C was replaced by U. You identify the affected codon. So it means if codons are not affected, there's no change, there's something else. Right? Describe which nucleotide has been replaced. Remember, we have spoken about that. You compare the original base sequence with the base sequence after mutation has taken place. You have discovered that, oh, C was replaced by U. Okay? You describe what was replaced. You describe what was deleted as well. So this is clear. And I made it even better for you to see it, that C was replaced by U. Okay, I hope this makes sense. Okay. The second scenario, this is the case scenario number 2. Remember, we are looking at the effect of mutation on protein structure. A protein is formed by amino acids bonded together by peptide bond, right? Those 20 can form one protein, right? Now, the second scenario, the table below shows the RNA codons that code for different amino acids. Remember, if after mutation has taken place and we've got different amino acids being formed, what does that tell you in terms of the structure of the DNA sequence? If after mutation has taken place and we've got different amino acids, what does it tell about the structure of the DNA sequence, right? So effect on mutation effect of mutation on protein structure, that is DNA sequence, case scenario 2. The table shows RNA codons that code for different amino acid. Then the DNA base triplet 1, 2, 3 below is read from left to right. These are the base triplet. Remember, I've spoken about the triplets, or you can refer them to codons. All right. So we've got GTC. This is the original base triplet. We've got GTC, we've got AAG, we've got CCT. Then mutation takes place. Remember, you are told, then you need to get, or you need to make application of what you were told, or what you are told in terms of what? Relate them to the questions that are given by the examiners. So this is the original strand. A mutation occurred which resulted in the following base sequence on the DNA molecule. So mutation takes place. Then let's observe after mutation has taken place, what happens. So we are told that on the first codon, we've got GTC, right? After mutation has taken place, what do we have? We have GTC. Good, they are the same, isn't it? Then on the second codon, we've got AAG, that is the original base sequence. But then on the second, after mutation has taken place, what do we have? A is replaced by T. Can you see that? A is replaced by T. Now you go to the third codon or the third triplet. We've got CCT, and then after mutation has taken place on the third triplet, we've got CCT. So the first and the last and the third, the first and the third codons compared to the base sequence after mutation has taken place, they are the same except for second codon. Can you see that? A is replaced by T. Right, describe the mutation that occurred. Yes, is gene mutation, sorry. And then steps that needs to be, or steps that are going to guide you, that are going to help you. Compare original strand base triplets to the one that has undergone a mutation, right? The steps that you need to take care of, right? So if you get it now, you won't struggle with your exam questions. You compare the original DNA base triplet, this is the one, right? Compare it to the one that has undergone mutation, that is, this one has undergone mutation. Can you see that? Then what do? Follow those steps. You identify the affected DNA base triplet and nucleotide. We have already identified those. And describe which nucleotide has been replaced or deleted. We have seen that. We have observed that. To say A was replaced by T. So steps 1, 2 are very, very important if you're given a scenario. So the answer is, remember you were to describe the mutation that occurred, right? In DNA base triplets 2, this one, the first adenine was replaced by T. A was replaced by T. Remember, you also need those symbols and you also need to know their names. So adenine was replaced by thymine. A was replaced by T. Okay, grade 12 learners. Right, now let's look at the questions. Remember, according to exam guideline, this is what you need to do. You need to understand transcription. What does it mean? You need all those steps. You need to understand translation as well. Question 2. Explain the effect that mutation will have on the resulting protein. Now, like I have explained to you, is that you will be given the data on the table. You don't need to worry about this. So this is mRNA codons, they are given there. Amino acids, they are given there. Right. Use the given table to find out if the new codon formed after mutation has taken place codes for the same or different amino acid. Now, if you compare amino acids, all these amino acids, for an example, if you look at the first amino acid, this one is serine. Okay, we've got all of those, 1, 2, 3, 4. The sixth one is also serine. What does it mean? We have, that is the original, and before and after mutation has taken place, it leads to same amino acids. So if we've got same amino acids being formed when my mutation has taken place, what does that mean? That should be the question that you ask yourself. If, for an example, is serine the first one and the second one, if you look at them, they are different. What does it mean? I gave both the scenarios. Right? You use the given table to find out if the new codon formed after mutation codes for the same or the different amino acid. All right? The affected codon AGC. Okay, can you see? This is AGC. In the original RNA codes for the amino acid serine. It's given. It's a given information. Right? Then the codon that has undergone mutation, which is AGU, that is when mutation has taken place, you go to the table and look for AGU. Remember, we looked for AGC, and AGC is serine as amino acid. And the codon that undergone mutation, AGU, where is AGU? You go to your mRNA codons, you identify that codon, AGU, this is AGU, right? Also codes for the same amino acid, that is serine. I hope this makes sense. So it's the given information, right? Now, what happens if the same amino acid is formed after mutation has taken place? It codes for the same amino acid, which is serine. The amino acid sequence will not change. The amino acid sequence will not change. Therefore, there will be no effect, or we are going to have same protein being formed. Why? Because after mutation has taken place, it resulted into what? Into the same amino acid. And if we have got the results of the same amino acid, this entails that there will be no effect, or we're going to have same protein being formed. A protein is formed by amino acids. Don't forget. Right? Going to the next question. Explain how this mutation will affect the protein that will be formed. Look at the steps. Use the given table to find out if the new codon formed after mutation codes for the same or different amino acid. So with that scenario, let's check what will happen. You first convert what? The DNA triplet of AAG to the mRNA codon UUC. AAG, remember A is replaced by U, or A matches with U, A with U, G with C. Then TAG. Okay, so convert the DNA triplet of bases AAG to mRNA codon UUC and TAG to the mRNA codon AUC. Look at this one. TAG, T, A, U. Remember, T is replaced by U. Then we've got GC before you could even read from the table. The codon UCC codes for amino acid phenylalanine, right? We've got the codon UUC, I'm sorry, which is phenylalanine, right? And then the codon AUC, which is AUC, this one, AUC codes for what? For amino acid isoleucine. This is the one. Now, in this scenario, we've got different amino acids being formed. And when different amino acids are being formed after mutation has taken place, this means that, listen, a different amino acid, there's two in this case, will be coded for instead of phenylalanine. The amino acid sequence will change, therefore a different protein may form. So I've indicated earlier on that when mutation has taken place, it doesn't mean we're going to have the result of a different protein all the time. So it will depend on the formation of the amino acid after mutation has taken place. All right, so I'm going to refer to the Gauteng Prelim Paper 2 of 2024. Later on throughout the year, we'll be looking at new questions. Remember, we're going to have June exam that is coming. So we're also going to refer to the end of the year 2024 papers to just tackle different samples of questions. Like I'm saying, you can't be getting same question year in, year out. So questions are going to change. You just need to study hard, know the content for all your topics. Remember, with life sciences, you'll be given tables, you'll be given graphs. So what is also important for all the topics that we'll be doing, you also need to know how to draw and label. Draw those structures, okay? So in this table, it shows a partial DNA sequence of a human. And table 2 shows the amino acid. Remember, we've got two tables, table 1 and table 2. That's the information that is given. It shows the amino acid coded for by different mRNA codons, okay? Now, it's important that when you answer questions, you look at the information given. Study the information first before you attempt. These are the base triplet 1, 2, 3, 4. These are our base triplets. And then we're going to, it's given that human DNA sequence. Triplet 1 is TGT, 2 ACG, 3 TGC, 4 ACA. So what is important also, give yourselves the mRNA or give yourself the anticodons of those codons before you can even attempt. That will also help you. I believe by now, because most of you are done with protein synthesis at school. I know most of you have started with meiosis already. So what needs to happen is for you, if you're given codon, to give yourself anticodons, right? Pre-planning for the questions that are going to be asked. Okay. So we've given mRNA codons. These are the codons given and the amino acids. All right. Let me go back. Right. Name and describe the process in protein synthesis that is responsible for formation of mRNA. Yes, we know that immediately when the question says name and describe, it means the name is going to be compulsory. You need to name that. So name the process. The process is what? Transcription. And describe the process in protein synthesis that is responsible for the formation of mRNA. I know Samantha have shown you how or where do we get a DNA. How does mRNA get formed, leaving the nucleus to the ribosome, you know? Okay, so name and describe the process in protein synthesis. Definitely you're going to get a compulsory name for the process. Describe the process. Now, in describing the process, you need to describe it the way it is from the exam guideline. If you are good at memorizing, you can memorize that, but with understanding so that you don't forget those key words. Describe the process in protein synthesis that is responsible for the formation of mRNA for 5 marks, meaning 1 name will be the name, 1 mark will be for the name, and 4 marks will be for the description of the process, right? So according to exam guideline, what is transcription? The double helix DNA unwinds. I just gave out 1 mark for that. The double helix DNA unwinds. Be careful. If you just mention that DNA unwinds, you're going to lose that 1 mark. You need to describe the shape of the DNA. Talk about the shape of the DNA throughout. So it is a double helix, as we have started with DNA code of life. So the structure of DNA is a double helix. So for transcription to take place, the double helix DNA unwinds. After unwinding, remember with the description, remember you are describing there. So description is not the same as naming, listing, is a bit of high order question. So when you describe, we are expecting you to at least show the statement there is in, to at least show the cause and the effect, right? So the double helix DNA unwinds, the double-stranded DNA unzips. Remember, because we have said is double helix, it has to unwind, unzip, okay? Or weak hydrogen bond break to form two separate strands. This is a mark. The double helix DNA unwinds, the double-stranded DNA unzips, or weak hydrogen bond breaks. Remember that nitrogenous bases are bonded together by hydrogen bond and not hydrogenous bond. It's a hydrogen bond. One strand, okay, weak hydrogen bonds break to form what? Two separate strands. This is very, very important again. So we need to see the logic as you're writing, as you are describing. Go back to your Bible, which is what? Exam guideline. Two strands are being formed. One strand. So it's going to be one strand, not each strand, just one of them. One strand is used as a template to form what? mRNA. Remember, with transcription, that's when we see the formation of what? mRNA, right? One strand is used as a template to form mRNA using free RNA nucleotide. This is very, very important. We need to see this. Using free nucleotides from the nucleoplasm. Can you see that? The mRNA is complementary to what? To the DNA. Okay, so be careful also, because we've indicated that the DNA, the RNA are written in caps. Be careful. You don't need to see the uppercase there for DNA and RNA. You are going to lose marks. Remember, you can't afford to lose those marks. You need that 100%, the 300 out of 300. Is possible? Right, we continue with Gauteng Prelim Paper 2 of 2024. Same information. These are the questions. Give the mRNA codon that is formed from base triplet number 4 on the DNA sequence. DNA sequence number 4, we've got ACA, right? Now give the mRNA codon that is formed for ACA. Remember, I've told you already you'll be having the anticodon here. Okay, right. ACU goes with what? UGU. Can you see that? A with, remember, T is replaced by U. Then C pairs with complements G. Then A with U, because T there is replaced by U. Name the amino acid coded for by the base triplet 2. You go to base triplet 2, right? Is ACG. You have the anticodon for this, and we know what is going to happen. We've got ACG. A will be U. Can you see that? C will be G. G will be C. So what happens is you write them so that when you refer to the follow-up question to that, you are ready to can just pick up easily. Now this is ACG, then it will become UGC, and the name of the amino acid is cysteine. Can you see that? Name the amino acid. You were just required to name the amino acid here. And the question is clear. You name it. Name the amino acid coded for by base triplet 2. You go to base triplet 2, you look at the codon, you get the anticodons for that, and it's UGC. The UGC, you go to your codons, you scan and look for that anticodon that matches the codon there. Then the name of the amino acid is cysteine. All right. A mutation causes base triplet 1. Remember, we are looking at the effect of mutation on the sequence of what? Protein being formed. Right? So, mutation causes triplet 1. Where is our triplet 1? Is TGD, okay? TGT, I'm sorry. Right, mutation causes base triplet 1 to change so that it looks the same as base triplet. This one, same, look the same as triplet 3. A mutation causes base triplet 1 to change so that it looks the same as base triplet 3. Explain what effect this mutation has on the protein being formed. And if you check, if you refer back to past papers, type of these questions like this one, the 4 marks, the 5 marks, they are of higher order. And you need to get the full marks. Remember, all of you are going to write the same paper. Whether it has got high order question or not, at the end of the day, each and every one of you is going to write the common paper. So you need to also practice those high order questions. So for 4 marks, explain what effect this mutation has on the protein being formed. Right? A mutation causes base triplet 1 to change so that it looks the same as base triplet 3, and then explain what effect this mutation has on the protein being formed for 4 marks. So remember, we're talking about TGT and TGC. So TGT, the one that is given in the information, will become TGC. Okay, remember, the question says, explain what effect this mutation has on the protein formed. Because they said mutation, after mutation has taken place, this one, TGD, becomes, you are told, it's in the information, it becomes, or they say mutation causes triplet 1 to change so that it looks the same as base triplet 3. So for them to look the same, look at how you need to approach that question. So the base triplet TGD, one is TGD, it will become TGC, which is 3. Therefore, the mRNA codon changes from ACA. How do we get the ACA? Remember, you look at this codon there, TGD, TGT. So T will go with A, G, C, T, A. This is an approach that you can use. And then it changes from ACA to ACG. How do we get the ACG? Remember, we've got T, G, C. So it will become ACG. All right. This is easy to go. So practice more of this high order questions. Then the protein will not be affected in this case. The same protein will be coded for. So let's look at ACA. Do we have ACA as mRNA codon on the table? Yes. Okay, ACA, right? We've got ACA, here is ACA. To change to ACG, do we have ACG? Yes. And if we look at ACG, this is ACG. If we look at the amino acid being formed, we've got the same amino acid, which is 309 for the 2 formed mRNA codons. Does it make sense, class? It should, right? Okay, so we're saying base triplet TGD will become TGC, then mRNA codon changes from ACA to ACG. I've shown you how to go about that one, which codes for the same amino acid 309. This is the one. Then we are going to have what? Same protein, right? The same protein will be coded for. KZN Prelim Paper 2 of 2024. Like I've mentioned that you need to have a practice of different papers. It can also be from different provinces, okay, for your practice. Maybe you can just take those set from 2021 up to the current year. Current year, it will be May 2025. Get those papers. If you're doing protein synthesis, you put all the sets there. You put all the sets there and check for those questions under protein synthesis, because you are there. As we progress on to other topics, you take the very same set of questions. You look at how examiners were asking those questions. How are you expected to approach those questions, right? KZN Prelim Paper 2 of 2024, pepsin, that is protein digesting enzyme. All right. Remember, proteins are enzymes in nature. Enzymes are proteins in nature, right? Protein digesting enzyme is a protein secreted in the stomach as inactive pepsinogen. That is the information given. In table 1, this is the table. Below, it shows the sequence of bases, sequence on the mRNA coding for some of the amino acids in a pepsin protein. Okay, we have got those. Number 1, we've got UUC2, AAG3, CUG4, UCG, 5, GAU, 6, AUC, 7, UGG. Okay, so in table 1 below shows the sequence of bases on the mRNA coding for some of the amino acids in a pepsin protein. Now, on what is given, look at those anticodons, right? Give the number of different anticodons needed to form this part of a pepsin protein. How many of those? Are you able to count? Now, as we were looking at all of those, the 7 of them, do you see any similarity there? Any difference there? You should ask yourself those questions. So, how many, give different anticodons needed to form this part of pepsin protein? We say they are 6. Why? Remember, you counting 1, 2, we counted 7 actually, but we're saying it's 6 because look at 1, 2, 3, 4, 5, 6, 7. But then this one, number 4 and 7 are the same. So we can't be repeating them. So these are the same. Therefore, we're going to have 1, 2, 3, 4, 5, 6. Hence, we are saying different anticodons here are needed to form this part of pepsin protein. Name the organelle where pepsin is synthesized in a human cell, right? Name the organelle where pepsin is synthesized in an organelle cell. We've got the ribosome there. Proteins are synthesized, or the site for protein synthesis is ribosome. So ribosome is the organelle where the formation of proteins takes place. Ribosomes are site for protein formation. KZN Prelim Paper 2 2024. This is the information given. Give the correct sequence of the tRNA base triplet, and if you can check, remember I've given you 2 scenarios, right? when amino acids are different, when amino acids are the same. So that should be the approach. Look at those codons, and get the mRNA there, anticodons for those. Give the correct sequence of the tRNA-based triplet that brings trypsin for the synthesis of pepsin. You look at the trypsin, and the trypsin there is what? It's AUG. Give the amino acid coded for by the mRNA-based triplet number 3. Base triplet number 3 is leucine, right? And then a mutation caused mRNA-based triplet number 6 to change from UUC to AUC. Explain the effect of mutation on the protein formed for 4 marks. We know different proteins will be formed. And when different proteins are formed, what happens? Since the anticodon AAG, right? AAG change to UAC, and the amino acid there is phenylalanine. It will be replaced by isoleucine. Then what we're saying is different proteins will be formed since this changed to UAG after mutation has taken place, and it will result in the amino acid phenylalanine that will be replaced by isoleucine, okay, in a protein, right? Describe the type of mutation referred to. Now, the type of mutation there is gene mutation. Why gene mutation? We know that gene mutation is nothing else. Remember, you also need to know your terms, right? So gene mutation is the change in the sequence of what? The bases, DNA sequence, nucleotides in a DNA molecule. Okay? DBE May June exam 2024. The diagram below represents a stage during synthesis of a certain protein. These questions are just repeated. Remember, I've given you a scenario of different questions. But then you take one, you can go with it, right? Now, this is what? Diagram representing a stage during synthesis of certain proteins, okay? Identify organelle 3. This is organelle 3. Now, organelle 3, that's where proteins are being formed. And what is the name of the organelle? Remember, when we started with the topic at the beginning of the year, that is in our first week, you had to take out the nucleus out of the cell, because you will be, for all these processes, you're going to be looking at some of the organelles there, like the ribosomes, like the nucleus. Describe the role of molecule 1. What is molecule 1? This is tRNA, isn't it? tRNA. Be mindful of the way you write tRNA. Describe the role of molecule 1 during this stage of protein synthesis. So it brings the required specific amino acid. Remember, if here we've got GGA, we cannot just having tRNA bringing just random anticodons. It should be specific. So the role of tRNA is to bring required or specific amino acid according to the mRNA or according to the codon to the ribosome. So tRNA brings required amino acid to the mRNA, right, class? Give the sequence of the nitrogenous bases. Sequence of nitrogenous bases there, we've got, remember, we've got GGA, then we're going to have CCU because T is replaced by U. On the DNA molecule that coded for histine, give the sequence of nitrogenous base, we have it there. B, on the DNA molecule that coded for histine, we have got GTA. This is histine, right? GTA. On the DNA molecule that coded for histine, you actually give the anticodon for that, right? During the synthesis of the same protein, there was a change in the amino acid sequence because alanine, where is alanine? The first one was replaced by tryptophan. Where is tryptophan? So this was replaced by this. Okay. Explain the possible reason for this. I'm finishing up. A mutation occurred that changed the sequence of nitrogenous bases of DNA from GGG to GAA when A was replaced by G. So you must talk about those things immediately when you are to explain, when you are to explain the possible reason for the change in the protein being formed. So A was replaced by G, and then mRNA codon, okay, it should be mRNA codon, change from CCC to CUU when U was replaced by C. This is the language you need to talk about when you talk about the protein being formed when mutation has taken place, causing tRNA to bring a different amino acid to causing tRNA to bring a different amino acid, right, class? Key points to remember. Now, as I conclude on the key points to remember, girls and boys, for DNA code of life. Remember, you started with revision of the cell up to protein synthesis. So these are the key points to remember. You need to remember that history of discovery of the DNA molecule. You need to know those scientists who discovered the history of the discovery of the DNA is Watson and Crick. We've got Franklin and Wilkins. So you can get those type of questions in your multiple choice question. Structural differences between DNA and RNA, you need to know that. That one you need to know. You need to also know the difference between DNA, mRNA, and tRNA. It is also important. Again, you need to know you need to be able to differentiate between DNA replication and protein synthesis. We have done this in the past or previous lessons. You need to know the difference between transcription and translation, and those must be learned by heart because it comes straight from the exam guideline. Also, you need to know the importance of DNA profile, right? For application question, importance, because we can give you those DNA profile for you profiling, DNA profiling. We can give you questions whereby you need to apply the knowledge or the content learned. You need to know paternity testing involves both parents and the child. When we talk about paternity testing, you must know that parents come in there, the child comes in. You need to know the uses and interpretation of DNA profiling. Okay, so in that way, you will score the weightings of this topic. You will get full marks when you look at those important points that I gave you. Okay, thank you. This is the end of our lesson for today. We will meet next time when we do meiosis, and good luck for the entire year. Study very, very hard because those that are doing well have started now. I know some of you started on the 6th of January. Some of you are 3 weeks into the content. Some of you are ahead. Remember, the exam guideline will also assist you to work ahead. Don't wait for the teacher. Work ahead. Thank you and good luck. Bye.